Good evening, everyone. Welcome you all to the Week 5 NPTEL TA session, which is on zinc finger nucleus technology. Please let me know if I am not audible or or if my screen is not visible to you. Okay. So today we will be discussing some questions on your lecture 5 series, and the questions will be basically based on zinc finger nucleus technology. Okay. Let's get started. So here is our first question. Which is the most common type of secondary structural element in proteins? Option 1, alpha helix, beta sheet, parallel beta sheet, or antiparallel beta sheet. Please try to answer this question in 2 minutes. Then we will discuss every part of this question in detail. And
Felix,
yeah, okay, please try to answer in chat box. Then only I can see who is answering and what. Okay, please try to answer in the chat box. Everyone, please try to answer this question in the chat box. Then we will discuss. Okay, I got two answers. That is alpha helix and it's correct. Okay, okay, let's discuss then. So, before going to the answer alpha helix, we need to understand what is this alpha helix, beta sheet, parallel beta sheet, anti-parallel beta sheet, or secondary structural element in protein. What is this primary, secondary, and tertiary and quaternary structures? Okay, so I guess you all know about all the structural components of protein, but still I want to brush up your memory regarding this. Okay, so let's see. So just a second. So here it is the primary structure of protein. That means a polypeptide chain. As you know, proteins are made up of amino acids. So these are basically amino acids bonded together by the peptide bond. Okay. It's also called amide linkage between two amino acids. Okay. So when these primary structures get folded, it leads to production of the secondary structures. It can be like this or like this. Okay. So alpha helix and beta sheet structures produced by hydrogen bonds forming within the polypeptide. So when the interlinking of this polypeptide happens, it leads to two types of secondary structures, that is alpha helix and beta sheet. Okay, so after that, this is the kind of 2D structures. If we move to the tertiary structures, it leads to a three-dimensional structure, which is leads to overall folding of the protein containing secondary structures. So secondary structure for protein folding leads to tertiary structure. This is the region of secondary structure alpha helix, and it is showing a region of secondary structure beta sheet, and then it a random coil. Okay, it is a schematic representation of primary, secondary, and tertiary structure of protein, not the actual representation. It's just a schematic representation. You can go to the actual representation, moving to the PDB IDs of each and every protein. Any protein you like, you can get it via protein data bank. It's a software, or it's a website where you can find out each and every protein of your like, and you can see the alpha helical structure, beta sheet structure, or random coil, or tertiary or quaternary structures also. Okay, and this is the quaternary structure. It is a multi-subunit complex where each subunit is a distinct polypeptide chain. That means multiple polypeptide chains get arranged to form a quaternary structures. A linear polypeptide chain fold into regular repeating structure units. The first of these structures they proposed is spiral or helical and named it as alpha helix. That they means the scientist. Okay. First, this proposed this spiral or helical, named it as alpha helix. Second, they proposed another structural motif and they named it as beta pleated sheets. Okay, so why there is alpha and beta? It's just the order of their discovery. It's just the order of their discovery that it is alpha helix and beta pleated sheets. Okay, and they constitute the elements of protein secondary structure along with loops, turns, and hairpins. Okay, next, what is alpha helix? It is the most common type of secondary structural element in proteins, as we have already discussed till now, and there are about 3.6 amino acid residues per turn in an alpha helix, or one residue every 100-degree rotation. This is a kind of fact you can remember for your questions like B section in NET LS exam. Okay, so 3.6 amino acids per turn, or for every 100-degree rotation. Each residue is translated 1.5 angstrom along the helix axis, and the pitch of a turn is 5.4 angstrom. Okay, that means a vertical distance of 5.4 angstrom between structurally equivalent atoms in a turn. That is called a pitch of a turn. And this rod-shaped alpha helix is stabilized by hydrogen bonds between NH and CO groups of the main chain. Okay, NH group, and I guess everyone knows the skeletal structure of amino acids. It contains one carboxylic acid group, one amino group, one R group, and one hydrogen. Okay, so this NH and CO, which forms the peptide bonds, also helps in forming hydrogen bonds during the alpha helix formation. I guess everyone is clear with alpha helix, how it is made up of, and number of amino acids per turn, and the pitch of the uh, alpha helix, that is 5.4 angstrom. These are quite facts, you can remember it. Okay, let's move to our next question. Okay, so here what we are doing is uh, basically we are talking about DNA binding proteins. Today's class is based on the zinc finger nuclease technology. So before moving to what is zinc finger nuclease, or how we can use it for genetic engineering, we need to understand what is this zinc finger domain. Okay, so before that we need to understand why we are studying it. So these are basically DNA binding proteins. That's why we just talked about the types of protein structures, that is primary, secondary, tertiary, and quaternary, and how it is made up of. Okay. So this DNA binding proteins have central role in all aspects of genetic activity within an organism, such as transcription. Transcription means it's simple, that is RNA formation from DNA template. Okay, mRNA formation. So here also we also know that a number of proteins have been engaged in transcription process. So these are DNA binding proteins. They also help in packaging like RNA splicing or other packaging systems. Rearrangement, that means genetic rearrangement. Replication, you all know that DNA replication, formation of a new DNA strand from the respective template strands. And repair, we have discussed a lot about DNA damage and repair, how it can be repaired by a number of proteins or enzymes. Okay, so how DNA binding proteins are helping us by helping us in transcription, packaging, rearrangement, replication, and repair. Okay, so a structural analysis of 240 protein DNA complexes, DNA binding proteins have yielded 8 different structural and functional groups. So, from a structural analysis of 240 protein DNA complexes, it has been found that they belong to almost 8 different structural or functional groups. Okay, which were further classified into 54 structural families. This protein structural or functional group can be divided into 8 types, that is helix torn helix, gene coordinating, zipper type, other alpha helix, beta sheet, beta hairpin or ribbon, other types, and enzyme. Okay, so today we will be talking about this gene coordinating domain, that means gene coordinating DNA binding protein. Understood? There are 8 types, and I encourage you to read about beta sheet, beta hairpin, or ribbon. Okay, I think everyone understood about this alpha helix. So we need to understand about the beta sheets also. But if I talk about beta sheet and beta hairpin, then it will be misleading our today's topic, that is zinc finger nucleus technology. That's why we'll be talking about gene coordinating DNA binding proteins today. Okay, let's move to our questions then. So this is our next question, which is zinc fingers were first identified in a study of transcription in the species African dwarf dog, clawed frog, threadfish, or wild dog. Which one it is? See, till now we have talked about that gene coordinating domains, they belong to DNA binding proteins. Okay, so that's why we are moving little by little through our slides, that what is zinc finger, what is its domain, and how it can be helpful in genetic engineering. Okay, I will be talking everything, so don't worry about it. Just try to answer this question, then we will discuss. Others also, please try to answer. Okay, I see every one of you answered B, African clawed frog. Okay, let's see. That means it's true, it's correct. Zinc fingers were first identified in a study of transcription in the African clawed frog, Genopus levis, in the laboratory of Aaron Cloak. He is the scientist who discovered it, Aaron Cloak. Okay, remember the scientist, he has a great contribution in this field. So now we need to talk about zinc finger domain. While I am talking about this, if you have any doubt regarding this, you can ask me in the chat box. Okay, I will clear your doubt regarding this topic. In 1940, biological role of zinc in proteins was established. Zinc is just a mineral. Okay, it was shown to participate in carbonic anhydrase catalytic activity. Soon it was found that zinc ions play a unique role not only for Ca carbonic anhydrase but also other enzymes catalytic activity. Okay, it is found that in 1940 that carbonic zinc is used, and it has important roles in other enzymes catalytic activity. In 1985, structural role of zinc was proposed for the Genopus levis African clawed frog transcription factor 3A, that is TF3A, in 1985. This is the biological role in 1940. In 1985, they found out the structural role of zinc using in a transcription factor 3A. The Genopus levis TF3A structure consists of repeating 30 amino acid residues folded around the zinc ion to form a small, highly independent domain called a finger. So why it is named as a finger? Because it look like it and contains 30 repeating amino acid residues folded around a zinc ion. Okay, and form a small, highly independent domain called finger. So here in the right side figure, you can see the structure of zinc finger domain. These are the beta strands and this is the alpha helix and it is the zinc ion Zn2 plus. Okay, so this is uh, all total forms the zinc finger domain. The structure of a typical zinc finger includes short two-stranded anti-parallel beta sheet. As you can see here, they are anti-parallel. Okay, I think no one have any questions regarding parallel and anti-parallel beta set. Okay, it's quite visible to everyone. It's anti-parallel beta set and an alpha helix. Okay, the whole structure here. This is the alpha helix and these two are anti-parallel beta sets. Two pair of conjugate histidine, this is one amino acid, and cysteine residues in the helix and beta strand coordinate a single zinc ion. So this whole thing can be called as a zinc finger domain. Okay, I think everyone is clear. And transcription factors often use zinc fingers to mediate protein DNA interaction. Got it? So one zinc finger domain contains two antiparallel beta strands, one alpha helix and one zinc ion. So now the question arises, when DNA protein interaction happens, is the zinc ion made any move? That means if it is true that the zinc ion is interacting with the negatively charged DNA, do you understand my question? I am asking if is it true that zinc ion interacts with the negatively charged DNA? That means it is positively charged zinc and DNA is negatively charged. So do you think they interact or not? So yeah, great. They don't interact. Here, the zinc finger domain, DNA binding proteins, alpha helix and beta strands and other amino acid involved in it, they interact, not the zinc ion. Okay. So if you face any questions like this, then try to remember this, that the zinc ion only used for the alpha helix and beta strand interaction, and this amino acids present in alpha helix and beta strands, they interact with the DNA and do the work, not the zinc ion. Okay, so I think everyone is clear with zinc finger domain. Okay, let's move to our next question. So till now we have discussed what is zinc finger. So please answer me. Zinc fingers have been classified into how many groups? 5, 8, 2 or 10? How many groups they have been classified? Please try to answer this question in 2 minutes. Then we will discuss. So everyone answered 8. That means 8 is the correct answer. That means zinc fingers has been have been divided or classified into 8 groups, and it is done by Krishna and associates. Okay, so let's discuss about the 8 folded groups of zinc finger domains. Okay, see, first one, just a second. First one is C2H2 like finger. The family members, for example, cis2H2 finger family, IAP domain family. Gag knuckle, retroviral gag knuckle family, polymerase gag knuckle, reovirus outer capsid protein gag knuckle. Treble clef finger, RING ring finger like, protein kinase cysteine rich domain. These are the family members of treble clef finger. Next is zinc ribbon, that is classical zinc ribbon or rubredoxin family. Next is zinc cysteine 6 like finger, and copper responsive transcription factors are the family members. Then TAZ2 domain like, it is the domain family. Then short zinc binding loops and metallothioneins. Okay, so generally C2H2 like finger, treble clef finger and the zinc ribbon comprise the majority of zinc fingers. So this C2H2 like finger and treble clef finger and zinc ribbon they comprise the whole zinc finger family. Okay, so If you want to remember this, then you can. Otherwise, we will be talking about this C2H2 like finger in our today's class. So it will be helpful for you and others also. You can find out the other things. Just a second. Yeah. So you can find out the other functions of the other family members also. Okay. So these are the 8 classes, and let's move to our next question. Which amino acids are conserved in C2H2 finger family? So just now we discussed the C2H2 finger family belongs to one of the zinc finger families. So options are cysteine and histidine, histidine and proline, lysine and arginine, aspartate and glutamate. Which one it is? Please try to answer this question. Then we will be discussing this in 2 minutes. Everyone, please try to answer. I guess this is a simple question because already the name suggests the name of the amino acids. Okay, I guess everyone answer. That is A. This means cysteine and histidine, right? So your answer is correct. As the name suggests, C2H2 means C for cysteine, H for histidine. This stands as the conjugated amino acid in C2H2 finger family. Okay, and cysteine has it is a semi-essential amino acid. It has a thiol group, SH group, and histidine it has one imidazole group. They both help in interacting with other amino acids and proteins and DNA. Okay, so let's talk about more C2H2 finger family. This C2H2 finger family, since its discovery in Genopus levis transcription factor 3A, the C2H2 zinc finger motif classic zinc finger, has been found to be present in many transcription factors and in other DNA binding proteins which recognize specific sequence of DNA. Okay, so this zinc finger domains or zinc finger family, they generally recognize a specific sequence of DNA, right? A typical C2H2 zinc finger contains a repeated 28 to 30 amino acid sequence, including two conserved cysteines and two conserved histidines. As the name suggests, C2 means two conserved cysteines, H2 means two conserved histidine. Okay, but other combination of cysteine and histidine as the zinc chelating residues are possible. The regulation of transcription seems to be the most important task performed by the C2H2 zinc fingers. So if anyone asks you question regarding this, that means if C2H2 zinc fingers generally associated with which work of the cell, that means it will be transcription regulation. Okay, just remember it in case you will find any question. However, recent findings suggest their roles in mediating protein-protein interaction. These are the PDB IDs. Okay, 1K2F, 1FV5, 1FU9. These are the PDB IDs, means protein data bank IDs. If you go for search for it, you will find out the structure of this zinc mediated protein-protein interaction. Okay, you can go for the PDB. Just search it. Just search PDB in your Google taskbar. You will be directed to the RCSB PDB's website, and from that you just enter this PDB IDs and you will find out this um, structure of this protein interacting with zinc. Okay, so I guess everyone is clear with C2H2 finger family of zinc finger domain. It is mostly helpful in regulating transcription. Okay, so here is our next question. The proposed sequential binding scheme of 3 C2H2 zinc finger domain TZD bound to the cognate DNA are specific binding, semi-specific binding, or non-specific binding. This means they are asking for the proposed scheme. That means which binding happens between C2H2 zinc finger domain and the cognate DNA. Read the options carefully and then answer it according to you. We will be discussing it in 2 minutes. Read these questions carefully, then answer. Everyone, please try to answer. Some people have joined recently, so please try to answer this question. We'll be discussing this question in 2 minutes. Okay, let's discuss this question. I have got lot of answers which say the answer is C. Okay, so let's see if it is correct or not, right? This is the slides I have taken from your lecture series, that is which shows the sequential binding scheme of TZD, that means 3 cis 2 H histidine 2 zinc finger domain to cognate DNA, and it generally says you from the slide only that it will be, sorry, non-specific binding, semi-specific binding, then specific binding. At first, let me say uh, what happens. This TZD, that means C2H2 zinc finger domain, they generally bind to the DNA in a non-specific manner. At any site, they just bind it. After that, it leads to semi-specific. They move to and they search for the specific DNA sequence where they need to be bound. Okay, search for the specific DNA sequence, that will be semi-specific binding, and then it leads to specific binding, then which is proper with the specific DNA sequence and TZT domain. Initially, TZT interacts with the cognate DNA in a non-specific binding. Then fingers 1 and 2 switch from non-specific binding to semi-specific binding. This is zinc finger 1, 2 and 3. They are colored in different colors, like zinc finger 1 is green, zinc finger 2 is blue and 3 is red. Okay, and this is the DNA. They just colored it differently for your clarification. With finger, first they interact in a non-specific manner, then fingers 1 and 2 switch from non-specific binding to semi-specific binding, with finger 1 almost fitting into the measure group completely, but finger 2 not fitting. Okay, finally the semi-specific binding is improved by the non-specific binding on finger 3, and DNA binding is then transformed to specific mode, with fingers 1 and 2 fitting into the measure group completely, but finger 3 domain non-specifically to DNA. So this basically talks about the TZD domain, that means C2H2 zinc finger domain. Here, what I said earlier, it generally stands for each and every zinc finger domain. That means at first they bind to the DNA in a non-specific manner at any site, and then they search for the specific DNS binding site. That means you can also understand this thing, that for example, earlier we talked about restriction enzymes and endonucleases, right? So they require a specific site for the cleavage of DNA. So, like that, zinc finger domains also require a specific site for their work, how to function at a particular DNA site. Okay, so at first they just bind at a non-specific site, and then they go for the specific sequence inside the DNA. Okay, it stands for all the zinc finger domains. But here we talk about it in a more molecular level, that is TZD. It's a name suggested, short form of TZD, zinc finger domain. At first, it is a non-specific binding. That means they just bind randomly and try to fit in the major groove and of DNA. So everyone knows about the major and minor groups of DNA, so I don't need to talk about it. So at first in a non-specific manner, then semi-specific. Semi-specific means one zinc finger domain, that is ZF1, it fits in the major group, but zinc finger 2, it can't fit. But in specific binding, this 1 and 2 domain both bound to the major group of DNA where they need to work or they need to function, and zinc finger 3 domain, it lacks it. It just stands there in a non-specific manner. Okay, so this is the molecular level of binding mechanism of C2H2 finger domain with their cognate DNA. Okay, I think everyone is clear. So let's move to our next question, which is, which restriction enzyme was used to make artificial ZFN, zinc finger nuclease? Please try to answer this question and we'll talk about it. So everyone has answered C, FOK1, right? So it's a correct answer. Great. So let's talk about ZFN, right? So earlier till now we have talked about zinc finger domain, how it binds to the DNA, at which manner in a molecular level also. So what is ZFN? It's zinc finger nuclease. Nuclease means which cleaves the nucleic acids or nucleotide, right? So as the name suggests, zinc finger nuclease is responsible for cutting the DNA. Okay. So in 1996, three scientists, namely Kim, Cha and Chandrasekharan, reported the deliberate creation of novel site-specific, this is site-specific endonuclease, by linking two different zinc finger proteins to the cleavage domain of FOK1 endonuclease. Okay. In our previous classes, we have talked about different types of endonuclease, exonuclease, restriction enzymes, and others. So I am briefing about it. Endonuclease means it cuts the DNA from inside. Endo means inside, inside the DNA. Okay, not at the 5 days end or 3 days end. If one nucleus cuts the nucleotides at a 5 days end or 3 days end of DNA, then it will be called as exonuclease. Okay. So FOK1 is an endonuclease which is combined with, just a second, FOK1 endonuclease cleavage domain has been combined to different zinc finger proteins, and it made it possible to create artificial nucleases that cuts DNA near a predetermined site. So basically what we want to do, we want to do targeted gene modification, right? This is our main motive for studying all this. So we want to go for genetic engineering. So that's why ZFN zinc finger nuclease is one important nuclease or enzyme which we can use artificially to cut the DNA at a specific site. Okay, so let's see. So how it came from basic research to translational research. Zinc finger nucleases resulted from basic research on zinc finger proteins and the FOK1 restriction enzyme. So restriction enzymes are enzymes that cuts DNA at a specific palindromic sequence. Okay. A ZFP zinc finger protein consisting of three linked zinc finger modules binds to cognate DNA site and carries out activities like transcription. Right? We have just talked about it in our previous slides, how zinc finger protein helps in transcription regulation. FOK1 has a bipartite structure with a separate DNA binding domain and a non-specific cleavage domain. So it has it has one bipartite structure which has separable. It is important. Okay, if it is non separable, that it can't be used for genetic engineering. It can be separable. That is one DNA binding domain, other non-specific cleavage domain. Okay, so scientists just combined these two and they made ZFN, zinc finger nuclease, are synthetic chimeric proteins composed of specific zinc finger DNA binding domain linked to a non-specific FOK1 domain or DNA endonuclease cleavage domain. Okay, I think everyone is clear with zinc finger nuclease. Okay, so for understanding the function of zinc finger nucleus and how it be helpful for genetic engineering, we need to understand what is zinc finger nucleus and how it was made artificially, right? So this is all. This zinc finger domain, it has specific sequence, and this cleavage domain, that is FOK1 DNA endonuclease, it has non-specific activity. If you have to face one tricky question, then it will be confusing for you. So please try to remember this. The zinc finger domains are specific and FOK1 endonuclease cuts in a non-specific manner. Okay, this cleavage domain. They just used the non-specific cleavage domain and separated the DNA binding domain and and replaced with zinc finger protein. I guess everyone is clear. So let's move to our next question then. Which is not the current technique used for gene delivery in ZFN? So please try to answer this question and we will discuss it in 2 minutes. Read the question carefully, read it again twice or thrice, how many times you need, and understand it and then answer. It's asking for not the current technique. Everyone, please try to read the question carefully and then answer. We will discuss it in 2 minutes. Okay, let's discuss. So the question is, which is not the current technique used for gene delivery in gene finger nuclease? Option 1, viral vectors like AAVS or ADVS, LVS, non-viral vectors like CPPs, physical methods containing microinjections and electroporation, etc. Fourth is GFP or molecular scissors. Some of you have answered C and some of you have answered D. Okay, so if you go through all the options, then it might quite clear for everyone, I guess, why it is misleading to some people. So see here, it is said GFP, that is green fluorescent protein, or molecular scissors. So how can molecular scissors can be used for gene delivery? It's quite simple, right? So molecular scissors, that means endonucleases, they have been used for cutting the nucleotides or DNA at specific sites or non-specific sites, as the name suggests, right? Molecular scissors. Scissor means cutting. It is used for cutting the DNA, right? So it can't be used for gene delivery in zinc finger nuclease. Simple. If someone don't know about all these vectors and methods, then also you can answer, right? Please try to understand the question, read the options twice or thrice if you want to understand it better. Okay, so I guess it's quite clear. Those who have answered C, I guess it's clear for you. Okay, if you have any doubt, then you can ask me. Okay, molecular scissors can't be used for gene delivery. Simple. So let's talk about others also. So current techniques used for gene delivery includes viral vectors containing adeno-associated viruses, adenovirus vectors, and lentivirus vectors. These are viral vectors. We have discussed about all these vectors in our previous classes while we talk about restriction enzymes and endonucleases. Okay. Non-viral vectors containing polymers, example polyethylene PEL, and others also like DO, TAP and CPPs, cell penetrating peptides. C. Physical methods containing microinjections and electroporation. This method, microinjection and electroporation, everyone knows about it because it is used generally for bacterial transfection, right? So when we transfer this our genetically modified protein or any kind of thing, you know, for E. coli also we have talked about microinjection and electroporation also, right? So this is the just a figure showing how these vectors can be helpful for delivering our ZFN, zinc finger nuclease, because it has been made outside of the cell, so it needs to be penetrated or moved to inside the cell for its functioning, right? So microinjection, this is electroporation, okay, and liposome, polymers, and AAVs. Got it? I think it's clear for everyone. Let's move to our next question. The CCR5 delta 32 mutation was initially discovered in 1996 as a genetic mutation that confers protection to cells from infection. Option A, coronavirus, HPV, HIV, smallpox. Please try to answer this question and we will discuss in 2 minutes. So everyone has answered C, HIV, right? So it's the correct answer. Great. So let's talk about what is CCR5 delta 32 mutation. This mutation was initially discovered in 1996 as a genetic mutation that confers protection to cells from infection by HIV. Okay, and this CCR5 delta 32 mutation, which was discovered over 20 years ago, disavers the CCR5 receptor on the surface of white blood cells. HIV uses this receptor almost like a key. It latches onto it to get into the cell. Without a working version of CCR5, HIV is essentially locked out of person's immune system. Okay, so what it is, HIV, as the name suggests. Okay, and it generally attacks the immune system of a person, and for this, CCR5 receptors, which is present at the surface of white blood cells, are important. They work as a receptor for moving the HIV into the white blood cells or person's immune system. When it is get mutated, and the mutation is named after CCR5 delta 32, then after the mutation it cannot be performing well as before, and it hampers the moving of HIV into immune system. Okay, so that's it. Let's move to our next question. Which of the following is famously known as the guardian of genome? I think everyone is familiar with this question. Yes, great. I'm getting answers. Everyone, please try to answer this question. I think it's quite easy for all the biologists everywhere. It's quite simple, right? So answer is P53. By stopping cells with mutated or damaged DNA from dividing, P53 helps prevent the development of tumors. Okay, it prevents the development of tumors. Because P53 is essential for regulating DNA repair and cell division, it has been nicknamed as guardian of genome. Okay, so guardian of genome. So earlier we talked about guardian of genome integrity. Do you remember, any of you? Anyone, do you remember guardian of genome integrity? Which is it? In our previous classes, we talked about it. Right, great, great. HR. That is homologous recombination. Great, thank you. Homologous recombination is named after guardian of genome integrity. But if you talk about guardian of genome, then it will be P53, right? Because it helps preventing the development of tumors by stopping the. Yes, someone trying to join. Yes. Okay. So this is the structure of P53 binding to DNA, the three-dimensional structure. Okay. Okay, then let's move to our next question. ZFNs were first used against dash. which prevented the virus from replicating within the host cell. HPV 1618, HPV E2 gene, HPV 16 E6 gene, and HPV P63. HPV stands for human papilloma virus. Please try to answer. Everyone, please try to answer. Okay. So I got two answers only. So answer is HPV E2 gene. Okay. HPV E2. Zinc finger nucleases were first used against HPV E2 gene, which prevented the virus from replicating within the host cell. Okay. So how this zinc finger nucleases work? This is the schematic representation of how this scheme ZFNs work. This is a intact DNA. ZFNs, zinc finger nucleases, talents, CRISPR-Cas9, they lead to targeted double-stranded break, as you can see here, right? So after becoming a after a breakage happens or damage happens to DNA, the system, the wild type DNA, they get the NHEJ, HR, they gets activated and lead to gene disruption by non-homologous and joining, gene correction by homologous recombination, transgene addition by homologous recombination. So we have talked about non-homologous end joining, homologous recombination in our previous classes. So as for our knowledge, we know that homologous recombination can be used for gene editing or gene modification, right? So this ZFNs, they what they do, they ZFNs generally form a double strand, as the name suggests, zinc finger nuclease. Nuclease means, ase means an enzyme which cleaves the nucleotides in DNA. Okay, so they just break the DNA, and after that, all the systems which repair, the repair mechanism inside the cell get activated, and they repair the double stranded break in by non-homologous end joining or homologous recombination or any type. Okay, so it depends upon the cell, not any artificial system interferes with the repair process. Okay, so don't be confused, they just induce one break at a particular specific site of DNA. Okay, where we want to induce some mutation or induce our specific sequence. Oh, we can't induce specific sequence, but yes, you can induce a mutation or disrupt the particular DNA. Fine, let's move to our next question then. This is a long question, please try to read it carefully once, twice, or thrice, then answer it correctly. Which of the following is incorrect in ZFN, means zinc finger nuclease mediated targeted genome modification via double strand break repair? Please read the options carefully. It's a long question and try to answer it. We will discuss into it 2 minutes. Here it asks for the incorrect one. Okay, which of the following is incorrect? Okay, let's discuss it then. ZFN-induced double-strand breaks are subject to cellular DNA repair processes that lead to both targeted mutagenesis and targeted gene replacement at remarkably high frequencies. Two discrete ZFNs recognize and bind to specific site at opposite DNA strands. Assembled fork-1 dimer specifically cleaves target DNA. Okay, so read it again. The options: a pair of zinc finger nucleases bind to a target sequence, dimeras through the fork1 domain and join the DNA at a defined state. It says join the DNA at a defined state. Next, the double strand break generated by zinc finger nuclease cleavage induces the DNA repair process. It's true, right? Because ZFN generally induces one double stranded break and that leads to DNA repair process. In the absence of donor template DNA, error-prone non-homologous end joining result in targeted mutagenesis. Okay, donor template DNA, why it is absent? Because it's generally results in say double-stranded break, not a single break in a single strand. Okay, so both the strands has been broken, so they can't be used as template for each other. Both the strands has been broken. Okay, so it leads to targeted mutagenesis. So it's correct. In the presence of homologous sequence, homology directed repair result in gene editing and site specific integration. So when there is homologous sequence, earlier we talked about here, okay, if there is homologous sequence, then it can lead to homologous homology repair or homologous recombination via holiday junction formation. Okay, so it's clear now what is the function of ZFN, how it mediate targeted genome modification. So simple, as we have already discussed, it induces double-stranded breaks and they lead to DNA repair process and targeted mutagenesis in case of non-homologous end joining and targeted gene replacement at remarkably high frequencies. Two discrete ZFN zinc finger nucleases. How they recognize at a specific sequence, opposite DNA strands, assembled by FOK1 dimer and cleaves the target DNA. Okay, I guess everyone is clear. So let's move to our next question then. Which of the following is a feature of DNA binding sites for regulatory proteins? Cystone, cytosine rich sequence, guanine rich sequence, CPG repeats, that means CPG island. You can understand this as you have heard about it. Palindromic sequence. Which one it will be? Please try to answer, everyone. Which one it will be? Okay, let's discuss then. Oh, I got only two answers. That is guanine rich sequence, cause but the answer is palindromic sequence, right? Generally, at the DNA binding sites of regulatory proteins, operators, promoters, and every cases, you will find palindromic sequences, right? It is a feature of DNA binding sites for regulatory proteins. The DNA binding sites for regulatory proteins are often inverted repeats of short DNA sequences. This DNA sequence is called a palindrome. Multiple subunits of a regulatory protein bind cooperatively at this site. So we have discussed about palindromic sequence. So I just need to brief about it. Palindrome sequences are those sequences of DNA which is read equally from 5 days to 3 days end or 3 days to 5 days end. If we want to read the sequence from 3 days to 5 days, then it will be same as when we read from 3 days to 5 days end. For example, one word is madam, M-A-D-A-M. If we read it from left to right, it will be madam. If we read it from right to left, then also it will be madam. So it is called one palindrome sequence. Okay. For example, I just gave you one example. It is one palindromic sequence. And in case of DNA binding sites, generally they present in the measure group of DNA, and uh, and they contain inverted repeats in both DNA strands, and they are short DNA sequences which is generally palindromic. Okay, let's move to our next question. What is the approximate length of DNA binding domains of regulatory proteins? Please try to answer. We have talked about regulatory proteins just now. So here is the question. What is the approximate length of DNA binding domains of regulatory proteins? Okay, let me discuss then. It is generally 60 to 90 amino acid residues. Okay, to interact with bases in major group of DNA, a protein requires relatively small substructure that can stably protrude from protein surface, right? Because generally this regulatory proteins which is responsible for transcription or any or any modifications, they fit into the major group of DNA, and for that we require a small amino acid sequence which is which can stably protrude from the protein surface and fit into the major group of DNA. Okay, and this is generally from 60 to 90 amino acids long. Okay, let's move to our next question. Zinc fingers do not have a role in which of the following processes? As we have already talked about, it is highly responsible for a lot of cellular processes. So, in which process it can't help the cell? Transcription regulation, ubiquitin mediated protein degradation, signal transduction, or cellular transport? Please try to answer this question. Okay, I got one answer only. So here is the answer. It is cellular transport. Okay. All the cases where DNA is not involved, right? Because zinc fingers are DNA binding motifs or DNA binding domains. So when in those cellular processes where DNA is not involved, that means it can't help the cell in those cases. Okay. So for cellular transport, zinc fingers don't have a role in this process. Whereas zinc fingers have a role in transcriptional regulation. We have talked about this C2H2 zinc finger domain has a lot of function in regulating transcription, ubiquitin mediated protein degradation and signal transcription. Moreover, it has a role in DNA repair, actin targeting, cell migration and various other processes also. Okay, so when the cellular processes don't involve DNA, that means zinc finger is not needed at that time. Let's move to our next question. This is the final question of today's session, so please try to answer. Zinc fingers can also function as which of the following: lipid binding motif, carbohydrate binding motif, ribosome binding motif, or RNA binding motif? Till now, we have talked about zinc fingers as a DNA binding motif. So, is it possible for zinc fingers to bind with lipids, carbohydrates, ribosomes, or RNA? Please try to answer this. Okay, let's discuss it then. So, it can act as a RNA binding motif. Okay, because we have discussed till now that zinc fingers are they require certain DNA sequences, or that means they are made up of nucleotides, right? Adenine, guanine, cytosine, or thymine. So it can act as RNA binding motif in lot of cases. Example, in certain proteins that bind eukaryotic mRNAs and act as translational repressors. Zinc fingers don't function as lipid binding motif, carbohydrate motif, or ribosome binding motif, right? So it has been proved that this zinc finger motif can bind to eukaryotic mRNA and act as translational repressors. Okay, so today we have talked about zinc finger domain, how it is made up of, and it is made up of generally two antiparallel beta sheets, one alpha helix and one zinc ion. Okay, so we discussed about how zinc finger nucleases has been made and how it has been helpful for uh, gene editing or targeted genome modification. And I think it will be helpful for uh, doing your assignments of this week. Okay, so you can read about it. I will highly suggest you to read your lectures, or you have given this lecture notes, right? So please go through the lecture notes so you will get a complete understanding of this topic, and you can do the questions of your assignments thoroughly. You can ask me this assignment doubts in our next class on Monday. Okay. So I want to ask if you have any doubt regarding today's session. Do you have any question? Do anyone of you have any questions regarding today's topic, or any feedback or suggestion? Okay then. Thank you so much for joining. We'll be discussing some questions or assignments in our next class, which is on next Monday. Okay.